Hello, Dr. Lloyd here for chapter 29. This is heredity. Okay, so for heredity, one of the major tests is called the karyotype. And what the scientists or technicians do is grow up certain cells and organize the chromosomes by size. Now you can see that they're pseudo-colored by location, but they're organized by size and they're numbered by size. So number one is the largest. Then there's 22 pairs of what's called autosomes, which are the somatic chromosomes, and then one pair of sex chromosomes. You can tell this person is a female because the X chromosome is quite larger than the Y. So this is called a karyotype, and it can be used to detect abnormalities like trisomies when you have three copies of a chromosome, or monosomies when you have one copy. And you can also look at major translocations that that might have occurred between chromosomes in case there was an error. Okay, so if we're looking at meiosis, production of the gametes, we had crossing over in prophase 1, and what we have here is metaphase 1 showed at the top frame of each cell. And before the gametes are made, these homologous chromosomes, let's say purple is from mom, green is from dad, these homologous chromosomes will independently assort at the metaphase plate. And so you can see that if we line up all the moms on one side, all the dads on the other, what we'll wind up with is an assortment of moms and dads' genes that go to a given gamete. And that's why it's really a random combination of mom and dad's genes that you get. Okay, so looking at homologous chromosomes, they've already gone through DNA replication, so you have two chromatids per chromosome, but there's only one centromere, so this is just a chromosome. It's kind of a double. Let's say these are number one chromosomes. They're all the largest ones, but they're not the X chromosome. During prophase 1, because they are homologous, or they contain the same genes, just maybe different form, a chiasma can form, or a cross, where genetic information is torn and exchanged between chromosomes. And this is a way where you can mix mom and dad's genes on the same chromosome through crossing over. And this happens in prophase 1 of meiosis. And then the gametes that are developed from these chromosomes will get a combination of crossing over or non-crossed over gametes from mom or dad. Okay, so the genotype is the letters or the form of the gene. The phenotype is the expressed characteristic. So let's look at albinism. This is a case where the skin is light colored, the hair is light colored, the eyes are usually light colored. And albinism results from a recessive or and so we can see that, let's say each gamete was heterozygous for this certain gene. In that case, you would develop a 1 to 2 to 1 ratio of capital A, 2 capital A lowercase a, and 1 lowercase a. So 1/4 of your offspring, and these are really the theoretical offspring. The actual offspring can vary, it can be any one of these four. There is a 25% chance of developing albinism when you have both recessive traits. There is a 20% chance having two capital A's, so that's the dominant non-albinism form, and a 50% chance of being heterozygous So once again, we've got gametogenesis going on, and we've got dad on the top, these are the sperm, mom on the sides, this is called a Punnett square, and let's say that they're both heterozygous. Hetero means different. Heterozygous means different forms of the gene, so this is a capital A, lowercase a. The capital A is what's called dominant. Lowercase a is recessive. Let's look at some conditions. Albinism was a recessively inherited condition, which means that you needed to have two copies of your recessive allele, no dominant alleles, in order for it to be expressed. Now, there's other conditions. The dominant gene can be the affected gene, or it could be the recessive gene. Let's look at some examples. So, syndactyly, or the combining of digits, usually webbed hands. This is actually a dominant phenotype, and the recessive is the normally expressed non-webbed fingers. Achondroplasia, this is dwarfism. It occurs in the heterozygous state, but it is a dominant condition, with the homozygous dominant being lethal. You have Huntington's disease. This is another dominant condition, and two copies of the recessive would be absence of Huntington's disease. Albinism, as we talked about, is a recessive gene, and so it needs two copies. Tay-Sachs, this is a neurological condition. This is a recessive condition, as well as cystic fibrosis. Now, we've talked about dominance and recessive, and this works for certain genes. But other groups, like blood group, the ABO blood groups, are not dominant and recessive, but are what's called co-dominant. So both alleles are expressed. So let's look at the phenotypes. So the phenotype is the expressed characteristic, and the genotype is the genetic characteristic. So let's look at type AB first. So we've got, looks like three different types of alleles. We've got I, well, I should say lowercase I, uppercase I, A, and uppercase I, B. So this corresponds to type O, which would be absence of A and absence of B, to type AB, which would be one copy of A and one copy of B. Now, if you're A, you could have two copies of A, or one copy of A and one of O, which would be absence of A and B. And if you're B, similarly, you can have two copies of B or just one copy of B. So blood groups are co-dominant. What that means is that there's a third phenotype where AB is mixed and it forms a different type of blood. And so the universal donors are O because they don't carry any of the AB antigens. Universal recipients are AB. They can receive O, they can receive A, they can receive B. And then A and B are combinations. Okay, here's a look at the sex chromosomes. This is the 23rd pair. This would be a male. We've got one X chromosome and one Y chromosome. And this is organized by a karyotype. And once again, 1 through 22 are the autosomes. And then 23rd pair is your sex chromosome. Now, conditions can be carried on the sex chromosomes or the somatic chromosomes. So if it's carried on the sex chromosomes, it's referred to as sex-linked. And let's look at a condition where we've got an X and a lowercase n, which would be the recessive allele. This female is heterozygous for what trait? This trait we're looking at is red-green color blindness. Now, in order to be colorblind, you need to have two copies of the colorblind gene, or just one copy if you're a male. Now, it's X-linked, which means it's carried on the X chromosome. And with X-linked conditions, you can be a carrier. Females are usually carriers, and the males are usually the affected ones. And this is because they just need one copy of red-green color blindness to be red-green colorblind. Females, because they have two X's, there's a decreased chance of being colorblind, but females are the carriers. So they're not affected because this is a recessive condition, and only those are affected are ones with the absence of the wild type or the capital. So once again, this is the Punnett square. We've got the male gametes, the sperm, on the top, and the female gametes, the eggs, on one side, and we're just multiplying these. Now, these gametes are all haploid, so these are the zygote of the potential offspring. So we've talked about recessive and dominant forms of conditions, but there's not just two forms of a gene. There could be many different forms, and this is the case with skin color. It's actually a polygene inheritance pattern, which means that there are multiple genes. Say that there are 3 genes. We can be able to explain if we mate somebody with very light skin with very dark skin, the first generation is going to be a mixture. And then this generation mates, and you get a spectrum of genetic expression or gene expression, leading to various shades of skin color from the very light to the very dark, with the frequency mostly being the mixture between the two. So this is a polygene type of inheritance. Skin color is a great example because it's definitely more than 2 genes. This is showing 3 genes, but it can be even more than that. Okay, this is a pedigree. So there are a couple different ways of testing for genetic conditions in families. There's doing the pedigree, which is looking at the generations of who has married who and had which kids. So let's look at the males are square, the females are circular. We've got a union here between a male and a female, and they have had 4 children, 1 daughter and 3 males. Now, this is a chondroplasia, which is a dominant mutation. And we can see if an affected individual marries a non-affected, half of their offspring will be affected. If 2 people with dwarfism mate, then there's a 50% possibility that they'll have dwarfism and 50% possibility that they will have normal endochondral ossification. So this is a pedigree. It's a great way of looking at families and affected individuals and whether the condition is dominant or recessive. Now, sampling the fetus can be accomplished in a couple different manners. One is to collect the amniotic fluid. This is done through what's called an amniocentesis. Usually it's guided by an ultrasound, so the physician knowing there is a risk of nicking the fetus. So this is the most invasive type of testing, is the amniocentesis. But it takes a couple of weeks to grow up these cells and get chromosomes into a karyotype. There's also a chorionic villi sampling, which is a little bit less invasive than the amniocentesis, but it still has a risk of causing birth defects and miscarriages. And so the chorionic villi sampling uh, uses a little bit of suction to grab some of this chorionic villi and separate the fetal cells from the maternal cells and grow those up and karyotype them. Okay, that concludes the chapter. See you in class.